Hi everyone, my name's Annalise, and let's jump into my presentation here. So here we go. I did my presentation on ancient humans and how they made a face for them. So this is a Denisovan female face that has been completely reconstructed without prior knowledge of what they looked like or even very good fossils or remains. So, who were the Denisovans? The Denisovans were cave dwellers. They primarily have had remains found in Siberia, and these remains were found alongside stone tools. They were dated, and the older tools were a little more rudimentary and basic, but they found younger tools, so more recent tools that were refined compared to the older tools. So they were, critically thinking and making their tools better, which is important in terms of ancient human evolution. They also found jewelry in these caves with the remains. And I just put genetic diversity here because it's interesting. They did have more genetic diversity than Neanderthals, and we'll get into that a little bit later here. This is a fun family tree that shows ancient humans when they lived and who else was living kind of at the same time here. So more recent, up here we have the Denisovans who lived about 800,000 to 150,000 years ago. They lived alongside Neanderthals, Homo sapiens, and Homo florensis were alive at the same time, although there was an interaction between Homo florensis and Denisovans here. There was interaction between these three groups, though, which we'll see here. So this family tree shows that, Denisovans, Homo sapiens, and Neanderthals all kind of came from Homo erectus, although we're not right sure here, the connection. And these little arrows show interbreeding. So there's interbreeding between Homo sapiens and Neanderthals and between Homo sapiens and Denisovans. Currently, Malaysians, was it native Australians and Eastern Asians have been found to have mitochondrial DNA that matches those of the Denisovans. There's also been a fossil found that is the first generation offspring of a Denisovan parent and a Neanderthal parent, and that's the first of its kind. We've found other fossil remains that have distant relations that were of different species, but this is the first one that shows direct crossbreeding between these two here. So that's partially why they have more genetic diversity, as I kind of mentioned on that previous slide. Um, but we didn't know what they looked like. So getting into the meat of it here, the problem was that there were very limited remains. So this here is a part of a mandible, not even a whole mandible. There were no complete skulls. They primarily just had like a couple of teeth and a finger bone or two. So really, really small remains. This is actually the largest remain here of an ancient Denisovan. So the question was, well, how are they going to determine what they look like? That's what everyone wants to know. And scientists decided to employ a method called DNA methylation analysis. So what is that? DNA methylation is found in DNA, RNA, and histones, and it regulates gene expression by making DNA either transcribable or not transcribable, thereby influencing the proteins being made, the phenotype being expressed, and the overall morphology of the species. Down here you can see unmethylated is not colored in, methylated is colored in. So these would be an unmethylated gene with the transcription factors, and voila, we get gene transcription and gene expression. Down here, if it is methylated, well, no transcription, no expression. And you see more DNA methylation activity during developmental years. So there's a lot of changes in DNA methylation. And that can also be influenced not just by development and hormones, but also by environmental influences like starvation, famine, that sort of thing. So how they use it. So they use loss of function mutations and DNA methylation to look at what proteins were being made, what phenotype was being built, and from there, what the morphology was. The next question is, well, how do we know that it's accurate? And what they did is they tested it on known profiles. So there are complete Neanderthal skulls. Obviously, we know what a chimpanzee looks like. They're still around today. And just by looking at the DNA methylation, they recreated these profiles. And by looking at what the known morphology was with the reconstructed morphology using DNA methylation, it was shown that it was pretty accurate. So we know that this is a fairly accurate method of reconstruction. And right here we have the Denisovan reconstructed profile. Just at a quick glance, I don't know if you can tell here, but this is a lot longer than the typical human arm. The Denisovans are also much stouter through their rib cage. But side by side, we can see here, so modern humans up here, Denisovans down here, and Neanderthals at the bottom. These are side-by-side comparisons of what the DNA methylation showed to be the differences between these three species. So with the modern humans, there's this kind of angle here. So that would be called facial protrusion. It's much more angled, whereas the Denisovan is much more upright, and Neanderthal is even more so. There's also cranial flatness. So humans have this kind of arched top. Denisovans, Neanderthals, much flatter. They found that Denisovans have thicker enamel, which is interesting as well. And I thought this was really cool, but look how thin the width is on the human mandible compared to the Denisovan and the Neanderthal mandible. It's much wider there. And overall, the face is much wider in a Denisovan, the widest in a Denisovan than the Neanderthal, and then the modern human. So this is the profile that they recreated so that we can do these sort of comparisons, because if we don't have skulls like these, or these reconstructed skulls, it's really hard to tell what they look like, compare them, and draw conclusions from there. So I put this picture back in here so you can see what the fully reconstructed face is. Other applications of this technology, well, we can determine the morphology of other extinct species and fossil remains, so we could make this whole spectrum of how ancient humans evolved and do more of those comparisons like we just did on the previous slide, which is really important. We can also compare gene expression linked diseases between humans and ancient humans. So if we know that, well, the methylation on a disease that is prevalent today is a problem, maybe the ancient humans had it too. Maybe they didn't. What could be the influences of that? Speaking of influences, though, that brings us down to limitations of this, is that we can't identify the specific environmental factors that had an influence on this morphology. So it's possible that we do another DNA methylation reconstruction and it looks really different than what we did, because we don't know how these influences interfere with the accuracy of what we're doing. But that's okay, we still have a bit of an idea, and like any other scientific endeavor, it's always important to remember our limitations. So that was my presentation there. Thank you guys so much. Feel free to ask questions, and I look forward to everyone else's.